So in order to utilize our cell-mediated immune response, the first thing we have to do is prime it. And just like a pump, you need to do things a little bit differently the first time around than the other times around. So when we're activating a naive T cell that has never before seen its antigen of interest for the very first time, it's going to have a different set of rules than it will once it's seen its antigen, is familiar with it, and knows how to find it. And this concept is called priming. It's similar to the term activating in that this is the first time our naive little T cell is going to grow up and see its antigen and get licensed to do what it's really supposed to do, to reach its full destiny. Now, recall from our lymphocyte circulation video that T cells are constantly surveilling the body, going in and out of lymph tissue and blood vessels. So this little T cell is going to come in through the afferent lymphatic duct and eventually traverse the high endothelial before it hangs out in the T cell zone, which is the paracortex of the lymph node. Just like the lymph node, antigens can come in through blood vessels or through the lymphatic duct and hang out in the lymph node, as can T cells and other things. Now, if this little antigen is hanging out near a T cell, it's not going to really do the T cell any good or us any good for getting rid of the pathogen because the T cell can't see it outside of the context of MHC. So lucky for us, there are dendritic cells that hang out within the lymph node, and also dendritic cells that may have picked up the pathogen out in the periphery and have brought it to the lymph node to be able to be recognized on MHC by the T cell after they have processed it. But how do we make this happen? How do we first have T cell and MHC meet? So let's go through the steps that are required to prime the cell-mediated immune response. And the first step is to create contact between the T cell and the antigen-presenting cell. So once inside the lymph node, the T cell needs to adhere to the dendritic cell before the TCR and the MHC can interact. So the first thing that's going to happen is that ICAM1 on the surface of the antigen-presenting cell is going to meet up with LFA1 on the surface of the T cell, like this. Note that the length of ICAM1 is fairly long, whereas the length of the TCR is fairly short. So ICAM1 with LFA1 helps kind of bring it into the docking station, which allows the MHC molecule and the TCR molecules to interact at their invariant chains. This interaction is also aided by CD4 and CD8, which, like ICAM1, are fairly long and will bring the MHC molecule in close contact, kind of locking that interaction into place. I liken this LFA1-ICAM1 interaction to shaking someone's hand when you first meet them, as opposed to giving them a giant big bear hug. At this point, the association between ICAM1 and LFA1 is fairly weak. It's just enough to bring the cells in close proximity so that the T cell can figure out if this is its antigen of interest. If the antigen within the MHC groove is the right antigen, the correct antigen for that TCR, that sends a signal to LFA1 that it's got the right antigen, and the interaction needs to get much stronger and quickly. So that will send a signal to LFA1 to actually change its conformation. This new conformation of LFA1 has a much stronger affinity for ICAM1, creating much stronger binding. As I mentioned, remember that the invariant sites of the MHC molecule will bind directly to the TCR molecule and CD4 or CD8 to the MHC molecule as well. This interaction here, MHC, TCR, and CD4 or CD8, is referred to as signal 1 of T cell activation. T cells require 2 signals for activation and a 3rd signal to differentiate into their full potential. The second signal of T cell activation is referred to as co-stimulation. T cells cannot be activated if there is no co-stimulation. It's kind of like a secondary control to say, nope, this is a real antigen, this is the real show, you found antigen, respond. This co-stimulation is mediated by the B7 molecules on the antigen-presenting cell. The B7 molecules are known as B7.1 and B7.2. They can also be known as CD80 or CD86. You might see them in both terms. Remember, immunologists are mean. We like to name things twice. But for the most part, I'll refer to them as the B7 molecules. B7 will interact with a molecule known as CD28 on the surface of the T cell. When this ligation occurs in the presence of the correct antigen with MHC and TCR interacting, you have signal 1 and now signal 2 of T cell activation. At this point, your T cell is activated. Now, what would happen if we had signal 1 over here with antigen and TCR and MHC, but no signal 2? Well, what happens then is that the cell becomes anergic. And I mentioned this earlier, that occasionally when cells are not activated properly, they become kind of like a zombie cells. They are unable to react, and even if they see their antigen of interest again later, in the presence of co-stimulation, they won't respond. The only way you can break energy is by giving them a lot of IL-2, because then they begin to get more signals that tell them, okay, maybe this is the real thing. But for the most part, they become completely unable to respond to that antigen in the future. Now, the addition of signal 2 will lead to two key events. The first would be the induction of the high-affinity IL-2 alpha receptor, which is known as CD25. Recall from our mechanisms of cytokine signaling video that all naive T cells express the gamma and beta chain of the IL-2 receptor, but that only activated T cells can express the IL-2 receptor alpha chain. When activated T cells upregulate the IL-2 receptor alpha chain, and especially if they are a CD4 positive T helper cell, they'll begin producing their own IL-2. As they produce their IL-2, they can then take up that IL-2, and that results in the cell proliferating. This is really important because we need T cell proliferation to create a nice vast army of T cells capable of combating the infection. Okay, so at this point, we have created contact between the T and the B cell. We created signal 1, which was MHC-TCR interaction, and signal 2, which is co-stimulation. Now we're going to add our last thing that we need to do, and we need to differentiate the T cell. This will be done using cytokines that are produced by the microenvironment and the antigen-presenting cell. At this point, the antigen-presenting cell will begin producing cytokines, or cytokines from the microenvironment, say other cells that have been activated, other antigen-presenting cells or T cells, will produce cytokines that will influence the type of T cell that will be differentiated. Even if it's a CD8 positive T cell, which doesn't have its own subsets other than the directive to kill target cells, these cytokines provide an important signal telling the CD8 positive T cell it's time to go to work. These cytokines include IL-6, IL-12, TGF-beta, and IL-4. Each of these cytokines, given in different concentrations or in different combinations, can result in different effector functions for the T cell if it's a CD4 positive T cell. And we'll cover how that works in another video looking at the different T helper subsets that exist. The junction between the antigen-presenting cell and the T cell, and this space in between with all of the various receptor-ligand interactions, is referred to as the immune synapse. The immune synapse, just like your neuronal synapse that you just learned about in your neuro studies, is really just the border between the two cells where the cells are able to communicate. So, in conclusion, when we're priming a naive T cell for the first time, we have 4 steps. You need to first create contact between the T cell and the antigen-presenting cell, and this is done through ICAM and LFA1, which then brings MHC and TCR in close proximity through the help of CD4 or CD8. MHC TCR binding is your signal 1 for activation, but that's not sufficient to activate a naive T cell. A naive T cell will also require co-stimulation, and that comes from CD28 on the T cell and B7 on the antigen presenting cell. Once this occurs, the CD4 positive T cell can begin making its own IL-2, and both CD4 and CD8 positive T cells will upregulate expression of the high affinity IL-2 receptor alpha chain, known as CD25. This signal will allow it to proliferate. Lastly, IL-2 plus a lot of other cytokines that I mentioned can promote the differentiation of the cell. For a CD8 positive T cell, it's going to license it to kill. For a CD4 positive T cell, it's going to license it to gain its true effector functions so that it can best help other cells clear pathogen.